Thank you for the opportunity to be here and to present growing evidence from pivotal clinical trials on self-amplifying mRNA vaccines on behalf of a pretty big group of researchers from Arcturus Therapeutics. Maybe I start a little bit about Arcturus. Arcturus is a late-stage global messenger RNA company which focused on development of prophylactic vaccines and significant opportunities in respiratory and liver rare disease. To accomplish the missions, company developed two platforms, which is LUNAR, a lipid-mediated multi-component nucleic acid delivery system with a library of about 250 proprietary lipids, and STAR technology platform, which combines self-replicating machinery with LUNAR into a single solution to produce protein inside the human body. The combinations of both platforms intend to induce robust and potentially superior versus conventional mRNA immune response and translate it into clinical benefit to the subjects. The company in clinical stage with 4 candidates is 2 for rare disease space for ornithine transcarbamylase deficiency and cystic fibrosis in phase 2 clinical study, as well as developing vaccines against influenza and COVID. And our COVID data set is probably the biggest today, which we would like to share with you. During the pandemic, Arcturus developed 5 COVID vaccines, starting from Arcturus 021, some kind of prototype vaccine, which include ancestral strain in native conformation. In parallel, company works in optimization of this strain, including double proline substitutions, foreign cleavage site modification, other changes to improve expression of the proteins. So in parallel with, during the outbreak of beta variant, the beta version of this vaccine was created. And more recently, to address recommendations of WHO and public health agencies, bivalent version of this vaccine and XBB-containing version of this vaccine was created. Overall, about 18,000 subjects was exposed to at least one of this formulation for the last 2 years, which we believe represents the biggest safety data set for self-amplifying vaccine generated so far. All these vaccines include 5 micrograms of total mRNA. We present primary results from 2 clinical trials. This is our first pivotal efficacy, safety, and immunogenicity studies, all studied during the pandemic. It's crazy study design with attempt to assess everything parallel in a single study. Today, we're just trying to present some efficacy results from 3B part of the study and some comparable immunogenicity data from part 3C. As you know, for demonstration of vaccine efficacy against clinical disease endpoints, it's very important to be in the right time and the right place. And Arcturus was lucky enough to start the study in Vietnam in August 2021, in the country which was on significant lockdown with 2% of population which was SARS-CoV exposed only, with 6 months before huge spike of Delta variant in the country, and in the situation when national immunization campaign was initiated to this have impact on the study by reduction of duration of follow-up, but also allowed to prevent significant mortality in placebo recipient, which was typical for the previous clinical studies. So in this study, vaccine efficacy against primary Delta variant of any severity was 56.6% with low bound of confidence interval 48, which satisfies study success criteria and also licensure criteria based on respective regulatory guidelines. Again, vaccine efficacy against CV COVID was 93.3% with 41 cases in placebo group and 2 cases in the study group, which also meet pre-specified success criteria and generated evidence of clinical protection induced by the vaccine. As we know, for COVID vaccine, it's extremely important to generate protection against the most vulnerable population of, including subjects with significant comorbidities associated with high risk of COVID and elderly population. And we were so happy to see the vaccine provide pretty consistent protection against all these groups for as prevention of vaccine of any severity and most important severe disease. Of course, we're trying to see on the persistence of antibodies, assuming the self-amplifying mRNA platform should provide some benefits compared to conventional vaccine and conventional mRNA vaccine. It's definitely a pretty difficult task during the pandemic due to natural immunity and hybrid immunity. In this slide, we try to stratify population to different groups, subject who doesn't have impact on hybrid immunity on the left sides. And in this case, you can see some kind of positive trend of antibody titers versus Vaxzevria in this case, with high immune response at day 57 and different kinetics beyond day 57. For individuals who have exposure to natural infection after vaccination, technically asymptomatic infection, they induce pretty robust immune response in the right part of this slide with technically no difference between vaccines regardless of primary vaccination schedule. Of course, it's a lot of expectations what will be reactogenicity profile of self-amplifying mRNA vaccine compared to conventional mRNA and compared to other vaccines. And here we present data for about 8,000 subjects per group for reactogenicity of the vaccine. You can see that local and systemic solicited adverse events with mainly mild and moderate. Proportion of severe reaction was 0.2%. A trend to low reactogenicity was observed after second and some subjects third vaccination. We have low frequency in elderly subjects consistent with other vaccines. And technically, a comparable safety profile was observed in subjects without comorbidities, regardless of gender, race, and other factors. Also, duration of the symptoms was pretty common for routine conventional vaccines, which we have majority of reactions within a couple of days post-vaccination, with mean duration of reactions of 2 days, and reduction of severity after the second vaccination. If you're looking about safety profile of this vaccine, we also don't see significant differences in the frequency of any reactions, any severe reactions, any related reactions after first and second vaccination. And if you're looking about medically significant, medically attended adverse events and serious adverse events, we have some kind of opposite trend. The frequency of reactions was lower in vaccinees compared to placebo group, which reflect reduction of COVID cases and associated adverse events in subjects who receive active vaccination. For the booster study, we conducted another clinical trials in collaboration with a Japan company, which is Meiji Seika, in last year. So this study recruited individuals who received 3 doses of Japan licensed mRNA vaccines, it's Comirnaty and Spikevax. And the subjects was randomized to receive second booster with Arcturus 154 candidate or Comirnaty. Both vaccine encoded ancestral strain. The study was conducted during the dominant circulation of Omicron BA.4/5 variant in Japan, and as such, study objectives include assessment of immunogenicity against ancestral strain and Omicron BA.4/5, with intention to show non-inferiority and, if lucky, superiority for self-amplifying mRNA vaccine. So the study was included more than 800 subjects. Majority was young adults. We have 80% of individuals who received 3 doses Comirnaty and 20% who received primary series of Spikevax and Comirnaty booster. Not surprisingly, all subjects have neutralizing antibodies before vaccination against Wuhan strain and 79 against Omicron variant. Here you can see results of the study. So the study compare immune response versus Comirnaty as measured by GMT ratio and difference in seroresponse rate. For GMT ratio for ancestral strain was 1.4 and low bound 126, which exceed pre-specified non-inferiority and superiority criteria. For seroresponse rate, difference was 13.6% with low bound 6.8 and also above both thresholds. Results for Omicron strain was a little bit lower, but also satisfied non-inferiority and superiority criteria. So when we presented this data to Lancet Infectious Disease, one of the reviewers say, okay, it's great, and what? So your comparator demonstrated great efficacy in hundreds of million people. You show 1.4 increase of antibody titers, how it's translated in clinical benefits. And I think these questions may be answered if you're looking about persistence of antibody titers following primary immunization, because you can see pretty common reduction of antibody titers after Comirnaty, which go into 30 to 50% of peak titers at 6 months post-vaccination, consistent with tons of publications. And you can see pretty plateau effect after self-amplifying mRNA vaccine between day 29 and day 91, and technically similar kinetic of waning after this time point. And as a result of this intergroup difference between groups at peak of immune response and subsequent time points is increasing. The study is ongoing. We hope to generate additional data to support this trend, hopefully up to 12 months after booster immunization. It was interesting to see how much this observation is consistent with other data. Here we present results from a relatively small study from Singapore, US, and South Africa, when we assess persistence following immunization of 3 vaccines developed by Arcturus, including 154 candidates. And you can see that GMFR remain pretty consistent within 12 months post-vaccination, which is consistent with data which we presented in the previous slide. Interesting that recently was at least a couple publications from phase 1 clinical trials from Gridstones and other vaccine developments who focus on self-amplifying vaccine, which technically go into very similar conclusion. What about tolerability profile of self-amplifying versus conventional mRNA vaccine? Again, we present data from, again, Japan study, which showing very similar rate of severe and any post-vaccination reactions for both groups. You can see maybe a little bit the difference for measurable local reactions, which twofold lower after self-amplifying vaccine, at least in this study. But in general, the safety profile of both vaccine and tolerability profile of both vaccines was highly comparable. In summary, so we believe that totality of available data allowed to conclude that self-amplifying platform for infection disease is well tolerated, scalable, with proven clinical efficacy, which may provide potential advantages compared to non-replicating vaccines in terms of superior immune response and potential to increase clinical protection, more durable immune response, and potential to require less frequent boosters, broad immune response, and potential to protect against immunological escape variants. And all these advantages was reached by significant reduction of total mRNA dose. As such, self-amplifying RNA platform create additional opportunities for development of vaccines against multiple viral and bacterial targets. And in conclusion, I would like to say thank you for researchers from Arcturus, CSL, Meiji Seika, Vietnam Biocare, as well as several academic institutions and investigators who contributed to individual studies and program overall. Thank you.
Thank you so much for the great talk. We have time for a couple of questions.
Yeah, thank you, Igor. Very good data. I wanted to ask you about the T-cell response. We know for COVID that T-cell probably play a critical role for protection against severity. Can you tell us a little bit about Arcturus platform in the ability to induce T-cells compared to more conventional mRNA platform?
No, thank you. It's a great question, because due to time limits, we don't focus too much on CMI response. We have data from early clinical studies when we justify the selection of dose, and we technically showed some pretty good Th1 directed immune response and some evidence of T-cell activations. We didn't generate data so far versus Comirnaty, technically one of the directions which we would like to go to characterize T-cell response and more specifically in the future.
Thank you. No more questions? Okay, one more question.
Hi, thanks for the talk, Florian Mann from Bayer. Can you comment a bit on your dose finding, and also did you measure protein levels versus standard mRNA vaccines?
Yeah, we performed those finding studies early in the pandemic using prototype vaccine 021, which have less immunogenic compared to candidate which we move in phase 2, phase 3 clinical trials. In these studies, we tested dose levels from 1 to 10 microgram, and we see some dose response between 1 and 5. I would say a very small increase between 5 and 7.5, and some increase of reactogenicity with previous construct with 10. So as such, based on the risk-benefit profile, 5 microgram was selected as a sufficient dose. So is it dose translatable to other pathogens and potentially to improved vaccine construct? It's difficult to say. It's definitely good fit for finally optimized 154 candidates, but we perform additional dose finding study with new target, for example, for influenza.
Thanks.
Thank you.
Hi, good morning. Miriam Mendila from CureVac. Thanks also from my side for the really great presentation. I have a question about the pivotal phase 3 study. You mentioned that at day 92 there was a switch over, so I assume a crossover from patients on the placebo arm to a real vaccine because of the implementation of a vaccination campaign. Did you analyze the impact on that crossover, like how many patients did crossover and how that impacted vaccine efficacy after day 92?
No, thank you. We definitely looking at the just compliance with crossover. It was surprisingly high because we have more than 90% of subjects who accepted crossover and received crossover vaccination. Of course, we try to assess efficacy after crossover, but you know it's extremely difficult because your both group was exposed with interval of 2-3 months, and it's very difficult to speculate about relative vaccine efficacy depending on time of vaccination. But again, compliance was extremely good, and technically we're happy that the study doesn't really cause any problem for placebo recipients who also vaccine crossover earlier.
Okay, thank you.